So COP2 and SAR, this is forward movement. Okay, according to the picture, we're moving from the ER to the urgent. These are proteins that must move on or onto the Golgi, or they're proteins that have accidentally escaped the ER. Okay. Sometimes this is called anti-angiography. I don't know why. Just call it forward movement. Well, because the backward movement is called retrograde movement. Let's just call it forward movement. Okay. We will spend more time, if we have time, with clathrin, because clathrin is classically understood the best in receptor-mediated endocytosis, which is still considered a part of the secretory pathway. Okay, it's okay to breathe? Okay, you are going to be encountering this, in this pathway, G proteins. We'll have G proteins up to wazoo in units 3 and 4, but we kind of, I want you to think about G proteins now, because some of these proteins are G proteins. Okay. G proteins are GTPases. They have enzymatic activity, okay? And they alternate between an inactive state and an active state. Can you think of some proteins we've already covered that are GTPases? Oh, did Kelsey have some? Yeah, but that's true. How about that we've covered, Kelsey said RAS, RAC, and RO, because that's, we've been talking about those in the biochem lab, but how about itself?
SRP.
Huh?
SRP.
SRP and the related protein, SRP receptor. Anything else? We looked at it in ER stress because it gets phosphorylated. EIF2, okay, is also a G protein. Okay, so the ones that Kelsey mentioned, RAS, RAC, and RO, belong to a different class of, kind of a unique class of G proteins. And we're going to be encountering a lot more in the next unit, and they also belong to a completely different class. So G proteins are extremely common in cell biology. Okay? They have to be activated. And activation requires, as we talked about before, pulling off the GDP, okay? That nucleotide gets removed from the G protein and is replaced with the GTP. It turns out that the SRP and the SRP receptor can do that job by itself. That's a unique feature of the SRP and the SRP receptor. Everybody, all the other G proteins require a guanine exchange factor. Okay, guanine exchange factor. So another protein that can pull the GDP off and replace it with GTP.
Which one can do it by itself?
SRP and SRP receptor, for some reason, doesn't need a guanine exchange factor because they can make that exchange by themselves.
So they can activate themselves? They can activate themselves.
Yeah, they can, and that means they have to get rid of the GDP and pick up a new GTP. I've seen on tests and papers before where some people think all you do is put a phosphate group on that GDP and make a GTP. Don't say that, because that's just dead wrong, okay? The whole nucleotide is taken off and a whole new nucleotide is put on. Okay? So we looked at, for example, EIF2, and it's EIF2B, it was its guanine exchange factor, if I remember correctly. Okay? All right. Some G proteins need some help with their activity. Okay? It is the G protein that hydrolyzes the GTP to GDP. But sometimes the activity level is so slow that they need to be urged, they need to be bumped a little bit. Okay? And the protein that can activate the G protein is GAP, for GTPase activating protein. Or you can call it let's call it GTPase activating protein. Okay? So the GAP is not the enzyme. It's not the enzyme that's going to hydrolyze the GTP. The G protein will hydrolyze its own GTP, but it might take a GAP to activate the enzymatic activity of the GTPase. Some proteins have their own inherent GTPase activity. They don't need any help at all. The SRP and SRP receptor don't need any GAP at all. Some of the G proteins we're going to encounter next unit don't need any help at all, but most do. Okay, so in this particular case, the proteins that you're going to see that have GTPase activity are SAR and R. So the GTP-bound SAR and ARF allow that coat formation. Okay, so we need this to get a coat to form and make a vesicle. In the GDP form, SAR and ARF cause the coats to come off the membrane. So this is like the reversal. You see the coats dissociate from the membrane. So this is coat association and coat dissociation. Okay. I was going to say, I will probably have this cycle, show you this cycle once again in the next unit to remind you that lots of proteins are G proteins. The cell uses actually very few strategies to regulate things, and this is one of the few strategies that the cell uses to regulate processes. Okay, so we got that. Okay, here comes a long, hairy process, okay? Here's where you might need about 8 colors. Okay, so I'm drawing the ER membrane. Okay, the cytoplasm's on top, the ER is on the lumen is on the bottom. This is a protein called SEC12. you only find Sec12 in the ER. So it must have the KKXX or KXKXX. It's the only place you find Sec12. This happens to be the guanine exchange factor for SAR, and only SAR. Okay? So let's make, I'm gonna make SAR yellow. So SAR in its inactive form is a cytoplasmic protein, okay? So was it made on ribosomes that stay floating in the cytoplasm, or membrane, or cytoplasms that associate with the ribosomes that associate with the ER?
Thank you.
It's a cytoplasmic protein, so it's going to be made on ribosomes that remain floating in the cytoplasm. Okay. So SAR can dock onto Sec12, and Sec12 will pull the GDP off, and as it pulls the GDP off, that creates a conformational change. In which a part of the SAR protein exposes some hydrophobic amino acids that allows it to insert into the ER membrane. Okay, and because Sec12 is only found in the ER membrane, that's the only membrane that SAR can insert in. It's the only location where SAR can get reactivated. Okay? So Sec12 will activate a lot of SARs.
I have a question.
Yeah.
When SAR binds to the Sec12, does it still have the GDP bound to it, or?
Yeah, it'll have, when SAR binds to Sec12, it'll still have its GDP on it, and then SAR will remove the GTP, and apparently this hydrophobic stretch of amino acids is exposed a little bit before the GTP gets put on. The sequence is not important, okay? All you need to know is that SAR with GDP bound it will bury its hydrophobic amino acids, and SAR with GT bound will expose the hydrophobic amino acid. Okay, so SAR with GTP is a membrane protein, and SAR with the GDP is not a membrane protein, it's a floater in the cytoplasm. Okay? But a bunch of these are going to accumulate. So I'm just going to draw, instead of writing SAR all the time, I will put GTP in the middle, okay? To remind you that this is a GTP bound form of SAR. Okay? Once it's in the ER membrane, SAR can recruit or become a docking site for a protein complex that was really called part of COP2. Okay? It's made up of this picture that I drew, this brick with holes in it, that I drew is actually two proteins. Sec23 and Sec24. Okay? They don't come to the ER membrane unless SAR is at the ER membrane. And I'm going to draw another one because these begin to accumulate. And you can go on, as the COP2 complexes form, they distort the membrane. Okay, so they're also curving the membrane at the same time they are forming. I think I will, if we have time at the end. Yeah. Oops, I shouldn't put GTP all the way. That's sorry. Okay. Sec 23 and Sec 24 happen to have different functions. Okay? Sec 23 is going to later on serve as the gap for SAR. Okay? It's going to activate the GTPase activity of SAR. Sec 24 is the cargo selection. And there are several, it turned out that there are several Sec 24s that bind randomly. Each kind of Sec 24 recognizes a different C-terminus. And we'll talk about that. Okay, so let's say we've been at it for, so let's build this and then I'm going to stop. Because this is going to get too out of control. I'm going to draw 3 complexes. Draw it smaller. And there we go. Okay, so now we have, as more and more COP2s join the scene, the membrane is forced to become distorted, and we see the beginning of a vesicle formation. Okay, the dent here that I haven't loaded up yet, this is where cargo binds. Okay, so we might have a protein whose job it is to bind luminal proteins. Okay, we've actually mentioned one already, and I'll remind you in a minute. Or we might have a protein that just needs to go on. Okay, maybe the insulin receptor or something that needs to just go on to the next destination.